Okay, so welcome to the lesson for today. We are going to look at mutations. We're going to look at mutations. We're going to specifically look at gene mutations. So we are going to realize that there are actually different types of mutations, which could be gene mutations and also can be chromosomal mutations. However, for our course, we're going to concentrate on what are known as gene mutations. Remember, if you can remember, when looking at the central dogma of molecular biology under translation, we did mention that a gene is a segment of DNA that codes for a complete peptide. And so when talking about gene mutations, we are talking about changes or alterations that can occur in an organism's DNA. So in short, a mutation is any permanent change in an organism's DNA. It is thus a modification of a cell's informative archive. It is a change in its genotype. Remember, you have the phenotype and the genotype. The genotype will determine the phenotype. What is visible is a phenotype. We have the genotype, which is modified in a case of mutation. So it means that mutations are going to create new alleles, and we're going to learn more about alleles when we start looking at genetics. I think after 2 or 3 lessons from now. So mutations create new alleles. And then we find that mutations can alter the DNA sequence, which can range from a single base pair to actually a whole set of chromosomes. So the mutation can happen in a single base pair to actually a whole set of chromosomes. Like I mentioned, the categories of mutations, there could be gene mutations and chromosomal mutations. So it can range just from a single base in a gene to the whole chromosome. Now remember the process, the central dogma of molecular biology. It has replication, DNA replicating, and that DNA can be transcribed, or rather is transcribed into messenger RNA, and then the messenger RNA is translated to form a protein. So on either of these processes, you're going to find that errors can arise, and hence these errors can lead to mutational changes in the gene. A gene is a segment of DNA which codes for a complete polypeptide. And so in the DNA, an error can occur during the replication process. During the process of transcription, an error can also occur within the RNA polymerase, which is going to lead to a mutation as well, or an error as mentioned earlier. And then even during the process of translation, the error can occur within the ribosome. And then errors can also occur during the process of folding, protein folding. Remember when we looked at proteins, we said that the shape of a protein, the folding of a protein is essential to its function. So we also find that actually errors can also occur within the protein when it is folding to its final shape. It means that any error that occurs, any error which occurs within these sources can lead to a non-functional polypeptide. Any error that can occur along this chain can lead to a non-functional protein or an error in the functioning of a protein. So a mutation can occur in a gene, and this mutation in the gene will affect the other processes of transcription, translation, and the folding of the protein. So these are some of the sources of error that occurs when you are looking at the central dogma of molecular biology. So gene mutation, we're going to concentrate on gene mutation. So categories of mutation. So they can broadly be categorized into whether they are gene mutation or they are chromosomal mutations, as mentioned earlier. And for our interest, we're going to focus on gene mutations, the gene mutations. So this is one of the categories. The other category that we see that mutations can be categorized in terms of the impact that they have on the organism, the impact they have on the organism. So they can be divided into 3 categories. So when a mutation happens, it's either that mutation is going to be beneficial to the organism. So I find that some mutations increase the fitness of the organism, meaning a certain mutation, when it happens, it enhances the ability of the organism to be able to survive and to be able to reproduce in certain environments. For example, we find that a mutation of G to A, guanine to adenine mutation, is actually beneficial in beach habitats because it renders the mice to camouflage. So there's this mutation of G to A in beach habitats, which renders certain mice to camouflage. So you find that this mutation is actually beneficial. And then the mutation can also be neutral. In a neutral mutation, it means that that mutation will have no effect. The mutation will render a non-effect. Yeah, so the mutation could be neutral. So if a mutation has no effect on the fitness, it is termed as neutral. So usually, as we are going to see, there are what are known as silent mutations. Usually these mutations are actually neutral. They have no effect. They are neither beneficial, they are neither deleterious. So the third category is that this mutation can be deleterious. They can be deleterious. So remember, because organisms tend to be well adapted to their current habitats, and because mutations are actually random changes that happen in the genotype, we find that many mutations lower the fitness of the organisms. So these mutations are what are known as harmful or deleterious mutations. Harmful or deleterious mutations. For example, I gave an example of the mutation of G to A, which is beneficial in the beach habitats because it camouflages mice. However, we find that this same G to A mutation would be deleterious or it would be harmful in the forest habitat. So this is another category. It can either be beneficial, neutral, or deleterious, or harmful. Another way of categorizing mutations is mutations can either be spontaneous or non-spontaneous. Mutations can either be spontaneous or non-spontaneous. So I find that these spontaneous ones are actually heritable. They are random changes which occurs in the base sequence of the DNA and could arise from natural phenomenon. They arise from the natural phenomenon. However, not all mutations occur spontaneously. We have what are known as mutagens, which can cause different types of mutations to occur. So these mutagens are the external physical and the chemical agents. And these mutagens include radiation, for example, X-rays, gamma rays, cosmic rays, and UV rays, and also certain chemicals. And we're going to further look at these mutagens in our next lesson. So mutagen can either be spontaneous or non-spontaneous. So when you look at the spontaneous one, they're going to occur from mistakes that happen during DNA replication or in defects during the process of mitotic or meiotic separation of chromosomes. So there are certain regions in DNA which are known as mutational hotspots. So these mutational hotspots are the parts which are most likely than others to undergo mutations. Are the ones that are likely to undergo mutations. So mutations, gene mutations can be spontaneous or induced or non-spontaneous. Okay, just a minute. Okay, so I was saying that these are some of the categories. They either be spontaneous or induced mutations. So now let's look at gene mutations. Let's look at gene mutations. So here we have a normal DNA sequence, a normal DNA strand. We have a normal DNA strand, and remember what will be transcribed will be the antisense strand. The antisense strand will be transcribed into messenger RNA, and the messenger RNA has a normal sequence here, and we have a normal sequence of the polypeptide. So this, the first diagram there, the first diagram on your top left describes the normal process. This is the normal. It has a normal template DNA, it has a normal messenger RNA, it has a normal polypeptide sequence for the primary structure. So there are different types of mutations that can occur. So we have a mutation which is known as base substitution. So by substitution, we mean that there's going to be a base will be replaced by another base. So the substitution of one base pair for another in DNA is what is known as a base substitution mutation. Now, of note is that when I looked at translation, we did talk about the genetic code being degenerate. Now, the degenerate nature of the genetic code would render base substitution to either being able to alter or not altering the amino acid sequence coded. So depending, depending on the mutation, it can either change the encoded amino acid, or it may not because of the degenerate nature of the genetic code. Now, if the new codon from the base substitution still encode the same amino acid, that type of mutation is what is known as the silent mutation, which is on your top right picture. Such a mutation is known as a silent mutation. As you can see, it's still going to code the same amino acid. So a silent mutation is that you have a base that has been replaced. However, the new codon from the base substitution still encodes the same amino acid. We then say that that mutation is a silent mutation. So a silent mutation or silent mutations are base pair substitutions that have no discernible effect. So as a given, or as already mentioned, a mutation in a protein coding gene that does not alter the amino acid sequence is what is known as a silent mutation, as in the example. It is a silent mutation. still going to code for the same amino acid. So in this case, the third amino acid, the amino acid will still be arginine because of the degenerate nature of the the genetic code. And then we have what are known as missense mutations. Missense mutations. So the base pair substitution, which results in a replacement of one amino acid by another, are what are known as the missense. So remember, we have the original sequence here. And in the original sequence, we have methionine, threonine, arginine, asparagine, tryptophan, and asparagine, or aspartate. So we have methionine, threonine, ARG, ASN, tryptophan, and AST. Now, the missense mutation, that base substitution, which has occurred, as in this case, which has occurred, as in this case, in which we find that one amino acid has been replaced by another, is what is known as the missense. Remember, the original sequence here we have arginine, but due to its base substitution sequence, base substitution mutation, what we have here is threonine. What we have is threonine. So this is what is known as a missense substitution. So it is called this same name of missense because the sense of the codon produced after transcription of the mutant gene would have been altered. So the gene has been altered, and hence it has lost the sense of that codon. Now, these missense mutations are grouped into two classes. So there are two classes of the missense mutations. They could either be what are known as transition or transitions mutations, and also transversions mutations. So transition mutations and transversions mutations. Now, when you talk about the transition does not change the type of base in the base pair. So in a transition missense mutation, that mutation does not change the type of bases in the pair. What I'm saying is that in a transition mutation, a pyrimidine will be substituted by a pyrimidine, or a purine will be substituted by a purine. So the transition mutation does not change the type of a base in the base pair that has been substituted. A pyrimidine will be substituted by a pyrimidine. A purine will be substituted by a purine. However, in contrast, when you look at the transversion mutation, the transversion mutation does change the type of base in a base pair. What I mean is that a pyrimidine will be changed to a purine, or a purine will be changed to a pyrimidine. Is that clear? Are we together so far? Is everything clear?
Yes, sir.
Okay, thank you very much. And then we have what is known as nonsense mutations. So this is a special category of base substitution, which arises when a base is changed. Now, when that base is changed, what happens is that the transcribed codon is converted to a stop codon. Very easy. a nonsense mutation arises when a base is changed such that the transcribed codon is converted to a stop codon. Remember, a stop codon, UAG is a stop codon. UAG is a stop codon. There has been a mutation that has occurred in the sequence. And this has rendered, during transcription, what has been transcribed is this codon. And UAG is a stop codon. And so such a mutation is what is known as a stop or a nonsense mutation. It's given this name of nonsense because it does not make sense. This mutation does not make sense to the translation apparatus. So now this stop codon is going to result in the premature termination of the translation, and it leads to a truncated polypeptide, a truncated protein. So I find that this sequence was supposed to be continuous. The translation was supposed to be continuous. Now, because of this nonsense substitution, base substitution, which has rendered this codon to be stop codon or a nonsense codon, it means that this translation process has been cut prematurely, and henceforth has led to a truncated protein. So in short, a nonsense mutations are base pair mutation that convert an amino acid specifying codon, an amino acid specifying codon was supposed to be tryptophan. Remember, we have made 309 ARG, ASN, then TRP. This has been converted to a stop codon. This is known as a nonsense mutation. It's known as a nonsense mutation. The nonsense mutation usually destroys the function of the gene product. It means that in the case of a protein specifying gene, the part of the polypeptide chain that follows the mutant stop codon here, supposed to be continuous, remember. Now it means that the part that was supposed to follow this mutant stop codon is actually missing. And this is deleterious. It means that the function of this protein is actually destroyed. The function of the protein is destroyed. And then we have what are known as frameshift mutations. Now, what are known as frameshift mutations. So frameshift mutations result from insertion or deleting of base pairs. So insertion or deletion of base pairs result in what is known as frameshift. So in short, when I talk about frameshift, there is either deleting or there is an addition of a single base. Now these actually have more profound consequences than the substitution of one base pair for another. These are called frameshift mutations because they're going to alter the reading frame in the messenger RNA downstream of the mutation. So now, when we change the reading frame in a gene, and thus we are also changing the messenger RNA transcript, it means that actually the majority of the protein that will be produced will actually be altered. Frameshift mutations can also cause premature termination of translation because 3 in 64 codons are stop codons, which represents a high probability in the sequence that has been randomized by the frameshift. So frameshift mutations can also lead to premature termination of translation. So during the frameshift mutation, as one base or as nucleotide bases are inserted into or as they are deleted from the molecule, resulting in the altering of the reading frame. What will happen is that the codons downstream of the insertion or the deletion are going to specify a whole different, they're going to specify a whole different sequence of new amino acid. Remember, our original sequence is this sequence. This is our original sequence. But because of this frameshift mutation that has occurred, we have a new sequence downstream of where the mutation has occurred, where the deletion has occurred, where the addition has occurred. We're going to have a new polypeptide, or rather a new sequence of amino acid. So depending on where the insertion or deletion has happened, there are different effects that are generated. As mentioned earlier, they can either generate a stop codon within a short distance of the mutation. A stop codon has been produced, leading to a premature termination of the translation process. And then I've already mentioned it also altering the amino acid sequence immediately after the change, as in the case on your bottom left, or bottom right rather, frameshift can change the sequence downstream of the mutation, or it can actually also code for a stop codon. So these are frameshift mutations. So these are the mutations that happens. So I've mentioned the substitution, base substitution mutations. We've mentioned the different types, and then we've mentioned also the frameshift, mutations. So frameshift, it's insertion or deletion of base pairs. Substitution, you are substituting one base for another. Okay, so those are the gene mutations. So we're not going to look at chromosomal mutations. Now, an important example of a gene mutation is sickle cell. It is sickle cell. It's a point mutation. It's actually a substitution mutation. Substitution mutation. So this is the normal sequence of DNA and RNA. What we have in the protein will be glutamate in the normal protein. But in the mutant protein, when there's sickle cell, when the hemoglobin is sickle cell, what will happen is that you are going to have valine. So there is a mutation that takes place in this. So the change of a single base pair, as we mentioned, can actually result in the changing of a single amino acid in a protein. And this can lead to a lot of debilitating clinical phenotype, or what can be seen, as in the case of sickle cell anemia. So in the sickle cell, we have a single A. We have a single A. We have an allele, a single A, an allele. This allele is changed to a T. Now, when the adenine is changed to the T, what results is a glutamic acid being replaced by valine. So the change of A to T results in glutamic acid being changed to a valine in the mutant. Now, this glutamic acid is actually a polar protein, or a polar amino acid rather. It's a polar amino acid. If you can recall from our lesson on amino acid, it's a polar amino acid. Whilst when you look at valine, it's a non-polar amino acid. Now, we did mention that hemoglobin actually contains 4 chains, 2 alpha chains and 2 beta chains. Now, the substitution of valine, of non-polar valine, causes the beta chains to aggregate into polymers. And this leads to alteration of the shape of the cells. And this leads to what is known as a sickle cell. The hemoglobin becomes sickle shaped. So as mentioned, the hemoglobin contains a tetramer of 2 alpha globin and 2 beta globin chains. So the sickle cell allele for beta globin gene contains a single base change, resulting in the substitution of valine for glutamine. This creates a hydrophobic region on the surface of the protein. And that hydrophobic region on the surface of the protein makes that protein to be sticky, leading to their association into long chains, which distort the shape of the red blood cells. So this is an important example of mutation, sickle cell disease, due to a mutation in the HB. So having said that, we come to the end of our lessons on mutation, and thank you for your participation. So the next topic that we're going to look at, mutagens. So we're going to meet tomorrow to look at the topic on mutagens.